both of those misapplied impeded drug discovery. And here's a couple quotes. There's one from Craig Venter here. The DNA reductionist viewpoint of the molecular genetics community has set drug discovery back by 10 to 15 years. Right? So, now, genomics is very, very important in drug discovery, but not the way it was hyped and not the way it was applied right around the year 2000. And here's a quote from me. In 1992 to 97, if you had stored combinatorial chemistry libraries in giant garbage dumpsters, you would have much improved drug discovery productivity. So in the earlier years, in the early 1990s, when chemists were making compounds with robots, the quality of the compounds was absolutely atrocious, and the properties of the compounds were atrocious, so much so that it would have been a miracle if anything useful had come out of it. Again, combinatorial chemistry has not disappeared, it's just grown up and become sophisticated. So those errors made in the early time frame have been fixed. Now, just to give you some idea, now, some of you may not realize how bad this got, but this genomics, HTS science madness, collaborations to mine genomic targets, massive HTS campaigns to discover ligands, 500 different targets, 1 million data points. The quote, this is a quote now, a wish to screen 100,000 compounds per day in a drug discovery factory and a wish to make a drug for each target, right? That actually came out, that was a quote from one of the major pharmaceutical companies, which has the initials GSK, so you know who it is. And here's a couple of quotes on here. So this was just madness. I mean, this hit close to home where I live. I live in Shoreline, Connecticut. So Bayer in West Haven had a collaboration with Millennium Pharmaceuticals, plowed multiple hundreds of millions of dollars into these genomic screens. This is a time era where people literally were emailing results from gene sequences to the US Patent Office. People actually thought that the target space would be wiped out by patents. Right? In retrospect, it was all crazy. The interesting thing about the Bayer is that although Bayer got absolutely nothing out of Millennium, as part of the deal, Bayer got an equity stake in Millennium, and Millennium stock went up to over $200 a share. So Bayer actually made a lot of money, but not on drug discovery, but they made it on the Millennium stock that they owned. And this is an example of, this is sort of right from the height of the madness, just pointing out that you couldn't make if you were discovering targets through a genomic approach, the success rates even back then were so absolutely horrible that you were, if that's all you did, you were guaranteed to go broke. You'd go bankrupt. Now, this is typically what pathways look like when you get started in a drug discovery project. When you don't know very much, everything looks simple and neat, right? And then what happens, the real sorry picture shows up, And unfortunately, you don't find out about this oftentimes until it's really too late. Howard showed the one slide where he was focusing on GSK3 beta. But at the bottom of the slide, he had P38 alpha, which is a very, very late stage. There have been over 20 clinical trials on that target, all of them a total bust. And now people are saying, well, gee, based on the clinical experience, maybe what we should have done is intercepted higher up in the signaling cascade. But it took this incredible expenditure of time, effort, so on, to find that out. It wasn't predictable a priori. Medicinal chemistry. 50 years of medicinal chemistry. So one of the things that's nice about this era, so I told you the chemistry is under control. One of the reasons it's under control is the pharmaceutical companies have really good records. And they're actually becoming pretty open in publishing those analyses. And they're really laying it out. What correlates with success? And so, for example, here's 50 years of medicinal chemistry. This is from a group at Vertex, and molecular weight goes up. So you might say, well, so what? Well, the problem is, the higher you go in molecular weight, the worse the success is in the clinic. So it actually is amazing that there's some very, very simple properties that correlate with clinical success. Attrition rates by phase. 2011, Nature Reviews Drug Discovery. Preclinical, phase 1, phase 2, phase 3, registration as a function of time. Right? It's getting worse. Every single phase, it's getting worse. What's really scary is phase 3, where it's really expensive. This is the steepest slope. So it used to be that, well, maybe a decade and a half ago, or for sure 2 decades ago, if you got into phase 3, 80% of those compounds will come out. Now, it's really pathetic, and it's continually getting worse. One of the common misconceptions, both on the chemistry side, medicinal chemistry side, and the biology side, is, gee, we really need a super potent nanomolar level or sub-nanomolar compound. This is an analysis by John Overington and Paul Gleason at National University of Singapore of oral drugs, marketed drugs. The average oral dose, 47 milligrams. Average IC50 is 5 times 10 to the minus 8, right? So 50 nanomolar. So no, not 5 nanomolar or 0.5. So to have success, you don't actually have to have a super potent compound. You just have to have everything else put together correctly. This is the slide Howard showed you. Again, it's this advantage of phenotypic screening. Now, this is one of the things, unfortunately, that's changed. Phenotypic screening was the way people did drug discovery in the 1970s, and then it was gradually supplanted by mechanistic screening. And if you look at the literature now, the trend is back towards phenotypic screening. So one of the really interesting things is that the new Translational Institute, the trend at NIH, 25% of the screens are reserved for phenotypic screening. So it's really nice to see. And this is what I just told you. So this is really good. And actually, okay, you're going to have a talk later on in this conference on natural products. I actually think that natural products are a really good fit for phenotypic screens. And I don't have time to talk about why that is, but there is a reason for that. So chemistry novelty is harmful. Okay. You won't see this in the literature. You will see it on the law side. This guy, who's a professor of patent law at Harvard, has written up about it. And he's really down on with respect to drug discovery. But there's a scientific, in addition to a legal argument. So at Pfizer, we used to have a grid for success in the clinic. And one of the criteria, one of the boxes was chemical structure. Was it similar to what we knew something about? So the more similar the new chemical structure was to things we'd worked on before, the higher the probability of success. So what does the patent system do? Because of composition of matter, it constantly moves us in the direction of novelty. And you see this in spades in kinases. People are just scrambling and pulling their hair out to come up with novel scaffolds. So every time a novel scaffold, you have to completely relearn over and over again everything about, the physical chemical properties, the ADME, the PK, the PD. You don't get a chance of capitalizing on what you've previously learned. It's a very, very bad system. Okay, there's no way I'm going to change it, but just to point out, scientifically, there is no logic, no reason to constantly trying to make new compounds, as opposed to making compounds that are variations of something you made before, where you're trying to breed in something useful. Screening diverse compounds is the worst way to discover a drug. Why is that? Because every publication I know of argues that biologically active compounds are not uniformly distributed through chemistry space. So again, I don't have time to go through the argument, but trust me, there's not a paper that I know of that says there's an even distribution of biologically active compounds through chemistry space. They're very, very highly clustered. Do drug structure networks map on biology networks? Very interesting work, been published in the last couple of years with some profound implications. So here I just cite to the paper by Brian Schoikert at the UCSF campus in Mission Bay. Okay. Now, you're probably all familiar with biology networks, right? So you construct networks of biology targets, usually based on something like primary sequence. Well, you can do something like this on the chemistry side, and you can say, I'm going to cluster compounds, drugs, by chemical similarity. Now, the problem is that chemical similarity can be described different ways. So Brian Schoikert did a lot of work in his publication to show that the network maps on the chemistry side were robust. And so what you can do is you can set up clusters of compounds related by chemical structure, and then you can annotate them by biological activity. Okay. Now here's a quote from the paper. A startling result from our initial work on pharmacological networks was the observation that networks based on ligand similarities differed greatly from those based on the sequence of identities among their targets. Biological targets may be related by their ligands, leading to connections unanticipated by bioinformatics similarities. Okay. Now, most of you have probably learned in class similarity in chemical structure equates to similarity in biology. If that were strictly true, then the biology networks and the chemistry networks ought to superimpose. But they don't. In fact, they don't superimpose very well at all. So what's the explanation for that? Well, the structure of the ligand reflects the target. That part is correct. But what happens is that evolution selects target structures to perform a useful biological function. So on the target side, on the biology side, there's absolutely nothing random about the targets. In fact, in a signaling network, there are very, very severe constraints that are placed on the structures of proteins. For example, some kind of a kinase network. The different forms of a protein, whether it's phosphorylated or not, have to be, the overall energetics have to be within 3 or 4 kilocalories of each other. And this happens all the way through, if you look at protein families, based on primary sequence, there could be unimaginable number of protein families. In point of fact, when you actually look at the structural work, there are only a relatively small number of protein families. And even within the ones that are known, the distribution is highly skewed. A very small number of protein families, fold families, account for the vast number of the protein structures that we know. Right? So because of this conservation on the target side, useful target structure is retained against the breadth of biology. So this happens, again, all the time. Through evolution, some useful function is found for a protein. That tends to be reused. Could be the exact same protein is reused, or you could have convergent evolution. So from different starting points, you come up with a protein structure that functions, even though the primary sequence may be different, from different viewpoints, you end up with proteins with the same function. So conservation in chemistry binding motifs really reflects the conservation in the target motifs. And also, again, people don't really talk too much about it, conservation in motifs where chemistry binding is not evolutionarily desired, you would also expect that. So if you think about this, in an evolutionary sense, there's absolutely no value whatsoever to having some kind of signaling cascade that can be messed up by some little molecule floating by. And so the structures have evolved to be robust, to not be perturbable. And and a lot of protein-protein interactions, fortunately not all of them, but will fall into this category of being very, very resistant to small molecule perturbation. So what's the implication? Right, so I throw this out to you for those either of you who have a chemistry background who work with chemists. So you have a screening hit. Structure-activity relationships on the historical chemistry of your hit can be useful even if it comes from a different biology area. Okay, so you discover, I have this compound, it's active in my new novel screen. What either you or one of your collaborators should do, you should go into the literature and find out, has anybody ever described structure-activity relationships on that compound? And don't worry at all about the biology. Don't pay any attention to it. Just see what has been done. Because what has been done historically in a completely different area may actually work in your new area. It sort of seems counterintuitive, but actually it's an explanation of a well-known medicinal chemistry management technique. When you're stuck in a project and you can't get it, you're making no progress at all, what do? You take a very experienced medicinal chemist who is uncontaminated by any of the thinking in the current project. You bring that person in, and he or she very often makes a breakthrough because they see something in the current chemistry and they say, 7 years ago I was working on something like this and I think the chemistry would work. And I know the biology is completely different, but I'm going to try it anyway. And it works. And it's this conservation of biology motifs translated into conservation of chemistry motifs that's the explanation for that. So the bottom line here is that here there's that medicinal chemistry due diligence is really essential. And to do this, you have to go into the chemistry side. You can't get this by going in the biology side. So just again, anybody who's in intellectual property and so on, it's very, very important. Cover the due diligence both on the chemistry and on the biology side. And I think I'm just about at the end here. So just to point out really the changes in drug discovery. I hope I've gotten the idea across that there's really a questioning of this reductionist approach. I really think that's a positive development in CNS drug discovery because I get a brick thrown at me, but I don't think a heck of a lot has come out in CNS drug discovery from the reductionist approach. I think a lot of it has come out from astute clinical observation and experimentation. And very few CNS agents are found rationally. Experimental observations in the clinic are extremely important. Okay, I'm impressed by multiple sclerosis as a paradigm. Eight approved drugs in this area, disease-modifying drugs. What happened? Why is this different than Alzheimer's disease? Well, back around a little over a decade ago, the technology got really good to have markers for disease progression. So MRI, a visual assay to pick up lesions in the optic nerve, some protein biomarkers in cerebral CSF. And the three together really allowed you to get an accurate fix on what the status of the patient was and was the disease progressing. And most important, if you intervened with some therapy, was it actually changing anything? Right? So really, I think it's very, very hard to get drugs until you really have some decent disease progression biomarkers. And I think that's probably my last slide, and I may have gone over a little bit of time. So thank you very much for your attention.